All right, on to translation now. Now that we've sent the instructions with the messenger, the mRNA, it's time to actually get the job done and make the protein. Okay, so we're going to do the second half of the central dogma, and that's translation. So, like I just said, you're going to take your mRNA from the nucleus and it's going to travel out of the nucleus through nuclear pores and it's going to be translated into a protein. So, that message is going to finally be turned into a physical embodiment. So after transcription, mRNA, like in the picture, leaves the nucleus through these little holes on the nuclear membrane called pores. And they're going to go find the ribosomes inside the cytoplasm. And that's going to actually be the mechanism or the little factory that's going to build the protein. During translation, you'll see that we tend to we tend to chunk our mRNA sequences into codons, which are 3 bases at a time. So 3 letters at a time is called a codon. Okay, so for here, we have adenine, we have cytosine, and guanine, and these 3 have been grouped together into one codon, and that's on mRNA. tRNA or transfer RNA have anticodons, so they exactly match. So here on our codon, we would have a U, G, C would be our anticodon that exactly is complementary to this codon. So codon, anticodon, they match, okay? And the anticodons are on tRNA, and when they match with the complementary codon on the mRNA, that tRNA has one specific amino acid attached to its other end. And that amino acid is going to get added onto the continually growing polypeptide chain that matches the mRNA. Okay, the chain is attached and created by the ribosome. So the ribosome is going to take the amino acid off of the tRNA after it's brought in based on the matching codons, and it's going to make that chain of amino acid connect to amino acid, connect to amino acid. And once you have a lot of amino acids together, you have a polypeptide or a protein. Okay, it'll keep growing and growing and growing until a codon on the mRNA actually signals for a stop. And there's 3 separate stop codons, as we'll see. So this is the shape of a tRNA. It's kind of like a lowercase t. And as you can see, there's an anticodon. So the codon that actually matches this on the mRNA would be a CGC codon. This anticodon is GCG, and it's attached to an arginine. Okay, that's just the name of one of the amino acids that exist. And there's 20 amino acids. And so an arginine will get attached to the growing chain of the protein. So like I said, there are 20 amino acids in total. There are multiple codons that match with one type of amino acid. So it's not like one codon is one amino acid. Otherwise, we'd have infinite numbers of codons and amino acids, right? But there are only 20 amino acids. Some amino acids have like 10 codons that make that amino acid, and some only have 1 or 2, right? Some are exclusive and some have a large variety. So like I said before, in mRNA, we have A, U, C, G, and those are the letters. And then in the words, we've grouped them into triplets or codons. So if you have an example here, you're going to have AAA, CGU, UCG, and CCC. And you want to group them into words or codons to make it easier for you to recognize and to kind of get ready to do the process of translation. So you would group it into AAA as a codon, CGU as a codon, UCG as a codon, and then the CCC as the last codon. And you're going to use a codon table chart for mRNA to actually produce the amino acid chain. And it's kind of cool to use. So I'll show you once we get to that. But in this case, AAA as a codon matches with an amino acid called lysine. CGU matches with arginine. UCG matches with serine. And CCC matches with proline. Okay. It's important for you to remember that when you have 3 nucleotides, you get 1 codon. And out of 1 codon, you get 1 amino acid. So if you have an mRNA that is 21 bases long, your amino acid chain is only going to be 7 amino acids because you divide it by 3, right? You're not going to get a 21 amino acid chain because the mRNA strand of 21 bases has to be broken into codons that have 3 at a time to be read for the protein. You can't just read an individual letter for a protein. You have to read a triplet or a codon at a time to get an amino acid to build that protein. So here's the big picture overall. So you have the central dogma where it goes from DNA, you're going to transcribe that to go into mRNA, and then you're going to translate it into protein. So if you can see over here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 bases on the DNA and the mRNA. But in the end, there's only 4 amino acids on this chain because you're dividing that 12 by 3 from the codon size. Okay, so here is our mRNA codon and amino acid chart. So this is the code that we're going to be using in order to get from our mRNA to actually the amino acid chain. Okay, so you're going to be reading a codon at a time. And the way it works is you're always going to be, it's like you're playing baseball. You're going to go from first base, second base to third base before you get a home run and you get your amino acid. So let's use a random codon, and we're going to use GCA. So our codon that we're going to use is GCA. So the first base is going to be the G. That means on this side where it says the first base, you're going to try to locate where the G is, and that goes UCAG. So it's going to be in this bottom row here. Okay. Second base, you're going to be going from the top now. So you're going to be looking for the second base, which is a C. So along the G's, there's going to be UCAG, which means that along the G, you're going to go to where the C is on the second base, and you're going to draw down here from the G and the C. Okay, that means we're going to be working in this box now. Okay, now the last or third base that we're going to look at is the A. So within this box, you want to see for the third base on this side, UCAG. We're going to look at G, C, and then in here, which one is GCA? GCA is this one right here. And that means that our amino acid is going to be ALA or alanine. Okay, so that's how you read it. You always go first base and second base and then third base. The more you practice, the better you get at it. And this is one of those things where in the beginning, it's like, what the heck is going on? Just remember first base, second base, third base. And once you get your actual amino acid, that's the home run. Okay, that's the home base. So let's do some more examples. So the other example that we're going to do is GUU. Okay, so GUU is going to be, let's do GUU. So we're going to start with G on first base. That means that we're going to be in this row, right? Because first base is this whole row. And then our second base is a U, which means that we're going to be looking at this column, and this column matches, this column matches with this row when it hits this box. Okay, and then in this box, we can find GUU, and that means we're going to have VAL as our amino acid, which means that on this page, we should be able to write VAL right here as our amino acid. So the next one we're going to do over here is CAU. Let's do CAU. Let me write it down here so I don't forget. CAU, first base is a C right here. So we're going to look at this row on the first base side with the C's. Our second base is an A, which means that the C and the C row and the A column match together in this box. And inside of this box, we're going to look at CAU. And CAU is up here. So it's going to be his or histamine. So we're going to be going up here, and we're going to want to write his right here. And we generally still shorten our amino acids to 3 letters as well, or a couple letters to kind of save space when we're writing the whole chain out. So the last triplet codon that we're going to do from mRNA is going to be AUG. So let's write it down over here so I don't forget it. And we're going to do AUG. So our first base is an A. So it's going to be in this row. U column in our second base is going to be this one. So A row, U column is going to be this box. The last one is AUG. And AUG down here is methionine or MET. Start is just the command. So usually at the beginning of mRNAs, you're going to have a methionine or a start code. You can also have it in the middle, but MET is the actual amino acid. You'll never write start, you'll write MET. Okay, so that means that on this page, we can write methionine in, which is going to be MET. And that is our answer there. So it's actually pretty straightforward. The more practice you get, the easier it gets and the faster you can do them. So you're going to make sure that you do your interactive notebook afterwards, which has some practice on this. And then there's an exit ticket as well, but it mostly goes over transcription.